we'll go into the details of insulin. So insulin is a 51 amino acid peptide, and it has two chains, the A chain and the B chain. And the chains are held together by interchain disulfide bonds. So you have interchain disulfide bonds and intrachain disulfide bonds. The interchain disulfide bonds are between two different chains, whereas intrachain disulfide bond means within the same chain between the amino acids, right? So for insulin, you have two disulfide bonds interchain between A and B, and you have one disulfide bond that is intrachain, that is within the A chain, right? So the insulin is actually synthesized in the inactive form as a pre-pro-insulin, as you can see in the figure that is given. So the signal sequence and an extra peptide is present in the pre-pro insulin, which is around 109 amino acids, almost double the number of amino acids required for insulin. And the pre-pro insulin is converted to pro-insulin by the removal of the signal peptide, which is almost 86 amino acids. The pro-insulin becomes almost 86 amino acids. And again, the pro-insulin is further cleaved into A and B peptides with the removal of the C peptide, which is a 35 amino acid segment. So the C peptide, it resembles the alphabet C, that's why it's called C peptide. So it removes the C peptide and you get the biologically active insulin, right? So the insulin secretion is dependent on plasma glucose concentration. So consider your plasma glucose concentration is very low. So when your plasma glucose concentration is low, the presence of membrane potential of the cells is approximately around minus 70 millivolts, as you already know. And because of the low glucose concentrations, there is very less glucose metabolism inside the cell. So when there is low glucose metabolism inside the cell, the ATP concentration is low. So when ATP is low, ADP is approximately or reciprocally high, right? Because ATP is converted to ADP in the process of energy release. So the ADP concentration is high. As a result, there is a opening of the potassium channels, and which allows the potassium, which is in higher concentration inside the cell, to be transported from inside of the cell to the outside of the cell, right? And this process does not cause the calcium channels to open, and as a result there is no insulin granule that is being released. So this is what happens in low plasma glucose concentration. So consider the situation where you have high plasma glucose concentration, and in high plasma glucose concentration, the glucose transporter that is present on the cell membrane, it acts as a sensor for the glucose concentration. It senses the presence of high glucose concentrations, allows glucose to be transported from the outside of the cell into the cell, and as a result, the glucose metabolism increases. When glucose metabolism increases, the concentration of ATP increases, concentration of ADP decreases. So when ATP-ADP ratio is high, the potassium channels are the potassium channels are closed, and these effects will lead to a depolarization of the membrane potential, leading to calcium channels to open. so that intracellular calcium concentrations are increased. So the intracellular calcium concentration allows the exocytosis of the secretory granules which contain insulin from the cell, so that insulin is released into the circulation. So this is what happens during high plasma glucose concentrations. So this is another figure which explains the whole process in detail. So consider the presence of glucose. You have the glucose transporter, that is GLUT. It can be GLUT1 or 2. And the GLUT1 allows the glucose to be transported into the cell where it undergoes glycolysis, where it takes part in the TCA cycle, and energy is produced. So protein, fatty acids, all of these are involved in the metabolic process so that the energy concentration increases and the ATP ADP ratio also increases. So when the ATP ADP ratio increases, it inhibits the potassium channels, but it activates depolarization, which will activate voltage gated calcium channels and allows the influx of sodium and calcium into the cells. So the influx of calcium is through both the type T and the type L calcium channels, and also the sodium concentration is also increasing inside the cell. So the intracellular calcium concentrations increase. At the same time, you have the extracellular or the membrane G protein coupled receptors that are present. Uh, Example is the acetylcholine receptor or the cholecystokinin receptor, or you have the GLP, GIP, glucagon receptor, and the acetylcholine cholecystokinin receptor is a G protein coupled receptor which activates the phospholipase C. So the phospholipase C converts phosphatidylinositol diphosphate into diacylglycerol and inositol triphosphate. So the diacylglycerol and inositol triphosphate in effect increases the calcium concentration. So the diacylglycerol activates PKC, protein kinase C, and the IP3 activates or initializes the endoplasmic reticulum to release the intracellular stores of calcium so that the calcium concentration increases, which activates the calmodulin kinase that is present. So the calcium concentration again increases in this case. Another factor is the glucagon receptor present on the cell surface, which is an adenyl cyclase activator, and the adenyl cyclase will synthesize cyclic AMP. The cyclic AMP will activate protein kinase A, and the protein kinase A further activates voltage dependent calcium channels, which will again increase the calcium concentration intracellularly. So the intracellular calcium concentrations will allow the fusion of the insulin containing secretory granules with with its exocytosis out of the cell. So these are the different processes that are affected by insulin. So it activates, the green shows activation and the red shows deactivation. So insulin increases glucose uptake, glycogen synthesis, lipogenesis, protein synthesis, gene expression, DNA synthesis, amino acid uptake, and sodium potassium pump. So all the cellular processes that require energy is activated with the help of the insulin, whereas all the systems which do not use energy but synthesize energy is inhibited or deregulated, so that gluconeogenesis, lipolysis, apoptosis, and autophagy is delimited or decreased in rate. So the interaction of the insulin and its receptor, and the receptor for insulin is a tyrosine kinase receptor, and The tyrosine kinase residues have IC domains that are phosphorylated. IC means intracellular domains which are phosphorylated, and the phosphorylation of this leads to the phosphorylation of serine on insulin receptor substrate 1, that is IRS1, and there are three categories of action that the receptor is important for. One is the PI3K activity or phosphatidylinositol triphosphate kinase activity. Second is the RAS MAP kinase ERK activity. And third is the activity that is independent of the IRS, which is usually uptake of glucose and the GLUT4 or the glucose transporter 4 movement. Alright.